Okay, good morning to everyone. Let's start our fourth session on neuroinflammation. This session was sponsored by Neuronal Signaling, and many thanks to them. We have slight change in our program because Diego was not able to come, and therefore we have another speaker, Peters and George-Hyslop. So please.
Good, thank you. So I've known Guy for an awful long time, probably longer than the average age of most people here. And so I know his facial gestures quite well. And when I arrived in the building yesterday morning, the look on his face was tantamount to that of a fox that's just broken into a chicken coop and is selecting a chicken for lunch. And it was clear that I was the chicken. But what I thought he was going to ask me for was a cell line or something like this, but that wasn't what he was asking for. And what he asked for is this emergency discussion today on some of the work that we've been doing and perhaps we can have my slides up, please. Good. Some of the work that we've been doing on risk genes that give either resilience or risk for Alzheimer's disease. And the particular gene that I'm going to talk about today is some studies on CD33, which we've been working on, and looking at the atomic structure of one of the splice forms and its relationship to Alzheimer's disease risk. And as you can see by the plethora of people involved here and the multiple institutions, this has actually been a project that's been ongoing for quite some time. And it has had a number of complexities, but I think I should be able to tell you something interesting in the next few minutes. But before I do that, let me just give you my statement of interest, which is very brief. I don't have any conflicts of interest, and I'm not talking about any medicines for off-label use. So the take-home points that I'd like to leave you with are that CD33 encodes an alternatively spliced sialic acid receptor, which is associated with risk for Alzheimer's disease. The splice forms are two forms. One is a longer isoform, and what I'm going to show you is that it forms dimers that are selectively trafficked to the cell surface, and that these long isoform dimers selectively bind one of two other AD-related molecules, actually two other AD-related molecules, clusterin and clusterin with A beta, and that this binding activates the immunoinhibitory signaling of CD33, which then reduces A beta uptake and amyloid plaque clearance. So in cartoon form, the central message is that this long isoform of CD33 forms these dimers. The dimers are trafficked to the cell surface where they can engage with clusterin or clusterin and A beta, and that this in turn then activates an inhibitory signaling pathway which shuts down basically some of the protective microglial responses. So, as I mentioned, CD33 encodes a sialic acid binding lectin receptor expressed on microglia and some forms of monocytes. It contains two isoforms arising from alternative splicing of exon 2, and exon 2 encodes the ligand binding site. So you have then two isoforms, and the relative splicing is modulated by non-coding risk alleles near CD33. And these risk alleles modulate the risk for Alzheimer's disease, and in the same direction, they modulate the absolute and relative expression of this long isoform. So the alleles that give you risk for Alzheimer's disease also increase the production of these long isoforms here. Subsequent studies after these GWAS studies had been done revealed that peripheral blood mononuclear cells from risk allele carriers who had more of the CD33 big M isoform had impaired uptake of A beta, and that amyloid PET scans done on the same individuals who were risk allele carriers and therefore had more CD33 big M had an increased brain amyloid burden. And together these observations suggested then that CD33 had an important role in modulating phagocytosis of A beta at least, and probably other irrelevant ligands as well. So the question was then, what was the structure of the ligand binding site? Because that might be useful in identifying potential inhibitors. And what were the actual ligands that were relevant to Alzheimer's disease that would bind to this ligand binding site? So to address this question, we purified the active domain, we crystallized it, we solved the three-dimensional structure of the active domain. And what we found was that this long isoform adopts a two-domain fold with two beta sheet sandwiches joined by a short linker domain in between these two things, and they are hinged at about 135 degrees. We looked at the binding site itself, and it was clear that it was very similar to the ligand binding site of most other Siglecs. It was sort of a flat planar subject, and it contained a cognate arginine at residue 119. The guanidino motif on the head of it was responsible for then interacting with the sialylated moieties of potential ligands, and that's a standard and cognate feature of most of these Siglecs. So unexpectedly, though, what we found was in the crystal structure that CD33 formed a dimer, as shown here, with the two ligand binding sites then slightly offset from each other at about 40 angstroms apart. And you can see in this rotational diagram how they sort of are offset from each other. And I'm going to sort of cartoon this in this way in the next few minutes. So the immediate question was, is this dimer actually just a crystal artifact, or is it real? And so to address this question, we undertook some biochemical studies, and the first of which was to look at the mobility of CD33 in non-denaturing blue native gels. And as you can see here, the migration pattern suggested that it was indeed an authentic dimer in cell lysates from a variety of cell types. The second thing that we did was that we transfected AGK293 cells either singly or doubly with two cDNAs, one of which was tagged with MYC and the other which was tagged with HA, and we then immunoprecipitated using the MYC tag, and in cells that we'd been transfected with two copies of the long isoform, you could see that the MYC, when you used it to pull down, would pull down the HA epitope. When you did the same thing in cells where you had just the small isoform, you could see that pulling down the small isoform pulled down the small HA-tagged equivalent, and in cells that were sort of heterozygous that contained both protein species, pulling down on the long isoform indeed pulled down the shorter isoform, indicating then that there was indeed a dimer formation. So subsequent studies done by us and by others has revealed that there's something rather interesting about these two protein isoforms. So it seems that the short isoform, even though it makes dimers, does not get to the cell surface and instead is trafficked out to peroxisomes. The same also happens to cells that are expressing both isoforms, where you have a mixed sort of heterozygous big M, little m dimer. But the species where you have a homodimer of the big isoform is trafficked to the cell surface, and we're able to show that using two different methods. So first of all, we used flow cytometry, and as you could see in cells that were expressing just the big isoform, you could see that the majority of cells were in fact had that big isoform at the cell surface. In cells which were expressing both isoforms, you could see that the predominant form of the protein that got to the cell surface was indeed still the big isoform. And in contrast, cells that were expressing just the smaller isoform, very little if it got to the cell surface at all. So the next question that we wanted to ask was, what are the ligands for dimeric CD33 big M in brain, and especially what are those ligands that are relevant to Alzheimer's disease? So we were aware of the fact that because it had these two ligand binding sites that were slightly geometrically offset, that this could open the potential for the binding of larger polysilylated ligands. But anyway, we started out with the usual methods, and what we were able to show was that indeed you could find a binding of ordinary sialylated sugars like sialolactose, but only with a rather modest binding affinity of 20 millimolar, which is not very impressive. We could have looked at sialylated lipids, but unlike Guy, who's a biochemist, when I had biochemistry in medical school, I simply went to sleep because lipid biology and biochemistry is coma-inducing, and so I just ignored it, and so I don't know anything about it, and so I couldn't think of any clever experiments to do. But we did have some ideas about what to look at in terms of soluble sialylated proteins, particularly big ones. And that was because there were two AD risk proteins, ApoE and clusterin, which are sialylated. And actually, clusterin is also known to interact with A-beta oligomers, and so that would be a third possibility that we could look at. So we looked at the binding of ApoE to clusterin, and we were not able to see any evidence of ApoE3 or ApoE4 binding to CD33. However, those same proteins were clearly able to bind to the LRP receptor, indicating that the protein itself was okay, they just don't bind to CD33. In contrast, when we looked at CD33, what we were able to show was that the big isoform was clearly capable of binding clusterin, as you can see in this IP here in the blue box. In contrast, when you expressed in those cells the little isoform, you could see that it did not interact with clusterin, as you would expect, since it doesn't have a sialic acid ligand binding site. Similarly, it was necessary that the clusterin be sialylated, so you could take ordinary sialylated clusterin, and you could do the same IP in cells that were expressing CD33 big M, and you could see that you could co-precipitate the clusterin with the CD33. In contrast, if you desialylated the clusterin, there was no binding, and again, that's what you would expect if the sialic acid moieties were important in this interaction rather than some non-specific protein-protein interaction. So we then conducted a number of studies to look at the avidity of this interaction using BLI and a couple of other methods, and they all came to roughly the same conclusion, that the binding affinity would be somewhere around 100 nanomolar, and that is very close to the estimated concentration of clusterin in the CSF, suggesting then that this might be an interaction which could occur in physiological circumstances. So the obvious next question was, well, that's a very nice test tube experiment, but does it actually happen in human brain? And to look at this, we used two orthogonal approaches, one of which was to look for optical evidence of co-localization of clusterin and CD33 in cells in brain using in situ proximity ligation assays, and the other was to document the presence of a physical interaction by co-immunoprecipitation assays. So we used this PLA assay to look for CD33 and CLU co-localization in microglia, and what we were able to see was that there was good evidence of this proximity ligation with lots of pixels of positivity for this interaction, and crucially, this showed a proximity relationship. So microglia that were closer to an amyloid plaque had more PLA pixels than did microglia that were far away from an amyloid plaque. And you can also look at this the other way, which is the way that Hyman and Spira-Jones has looked at, which is there seems to be a sort of a cutoff around 20 nanometers from a plaque, and what you could, 20 micrometers from a plaque, and what you could see was that those microglia that were close to a plaque had lots of PLA, and those that were further away had less PLA. Again, arguing then that there is a proximity relationship between the plaque, the amount of clusterin that was probably coming off, and A-beta that was coming off that plaque in that penumbra around the plaque, and the activation of nearby microglia. So the next thing we looked at was to see whether or not you could actually show a physical interaction between clusterin and CD33 in the brains of AD patients. And what we were able to show was that in control individuals, you could precipitate the CD33M, and even though there was clusterin in that brain, it wasn't close enough to the CD33 in order to get an interaction. In contrast, in individuals with Alzheimer's disease, with lots of amyloid plaques and presumably microglia close to those plaques and clustering around the plaques, we were clearly able to co-precipitate the clustering with the CD33 as shown and quantified here. So the next question we wanted to ask was, does clusterin and CD33M binding activate the immunomodulatory tyrosine-based inhibitory signaling pathways? And to address this, we used two approaches. One was to ask, does clusterin and CD33 binding actually activate that biochemical signaling pathway? And secondly, does that activation of that pathway have a functional effect on the things that had previously been shown relevant, namely the uptake of A-beta by human peripheral blood cells and by amyloid plaque clearance from brain. And the item signaling pathway is relatively well understood. So ligand binds to clusterin, activates then presumably a conformational change in the intracellular domain, which recruits SARC and then the SHP1 phosphatase, and this in turn then shuts down the signaling pathways that support the protective effects. So as you can see here, if you took just clusterin alone and you exposed it to U937 cells and then looked at the phosphorylation state of CD33M, you could see it became phosphorylated and it recruited SHP. In contrast, if you added A-beta as well, so premixed the A-beta with the clusterin and then put that on together, you actually got a much better activation of the signaling pathway. And this wasn't due to any change in the binding avidity. It appeared to be just some conformational change, which just makes it a better ligand without changing the binding affinity. So we then asked, does activation of that pathway actually occur in peripheral blood mononuclear cells? And to look at this, we then looked at the phosphorylation of CD33 in cells from donors who were CD33 big M positive. And what you're able to see is that there's a certain amount of basal phosphorylation, but if you give them clusterin, you can see that there is a dramatic and significant upregulation of that phosphorylation event. So this occurs not only in artificial preparations, but also in endogenous signaling pathways in real human peripheral blood mononuclear cells. So the final question that we wanted to ask was, does clusterin and CD33M binding have a functional impact on AD-relevant phenotypes like A-beta uptake and amyloid plaque removal? And again, I remind you that Betsy Bradshaw and colleagues and others had shown that peripheral blood mononuclear cells from risk allele carriers have an impaired uptake of A-beta, and PET scans done on those same individuals showed that there was an increased brain amyloid burden, indicating then, as I'd said previously, that this suggested that CD33M has some role in the phagocytosis of A-beta and obviously possibly other potential ligands as well. So in order to replicate this, we took the PBMCs from risk allele carriers and we measured the basal state, and then we looked at what happened when you administered clusterin, and as you can see, it reduced, the presence of clusterin reduced the uptake of A-beta in these cells. In contrast, if you used the same assay, cells from the same individuals, you could see that decellular clusterin, which doesn't bind to the CD33, failed to induce an impairment in A-beta uptake. So that supported that idea. So the next and final question was to ask whether or not this had any relevance in clearing amyloid plaques from brain slices. Our American colleagues who have bags of money are able to do fancy things like PET scans in mice, but poor Cambridge academics, we have to use a bit more brain power and a little bit of begging. And so Dan Frankel and Zvi Fischelson in Tel Aviv had a rather neat assay in which they looked at frozen brain sections from a transgenic mouse in which they put mononuclear cells on it and could show phagocytosis of these amyloid plaques. So we begged them to help us with this assay, and they kindly agreed. And what we were able to show was that in individuals, in peripheral blood mononuclear cells from individuals who had the protective AA allele, who were therefore expressing predominantly little m CD33, you could see that there was a significant reduction in the amyloid plaque burden measured by various different methods. In contrast, when you did the same thing with PBMCs from risk allele carriers, you could see that these risk allele carriers were unable to clear the plaques as well as the AA allele carriers. Again, being essentially the analogous result for what could have been done in vivo in humans with the amyloid PET scans. So the assay seems to be robust. So we then asked whether co-culturing human monocytes with clusterin on these brain slices would have an impact on the brain clearance. So we took initially just medium only, and we counted the plaques, and you can see that there's a certain abundance of these plaques as measured by, again, different ways of measuring the amyloid plaques. If you then took the cells and you incubated them with decylated clusterin, which doesn't have any effect. So the green bars are nothing. The red boxes are now cells with decylated clusterin, and you can see that the cells cleared the amyloid plaques quite substantially. If you then did the same thing with the same cells, but instead of using decylated clusterin, you used actual clusterin itself, and you could see that that significantly attenuated the clearance of the amyloid plaques, putting it almost back to the level that you'd see in preparations with no cells at all. So let me conclude then by pointing out, first of all, that CD33 big M isoform expression is regulated by these non-coding risk alleles that are associated with AD. If you have the risk allele, you have relatively more of this big isoform. This risk-enhanced CD33 big isoform has an intact ligand binding site, and the little isoform with the protective allele lacks that ligand binding site. Both forms form these physiological dimers, but only the large form gets to the cell surface, and there it's able to bind various ligands, including clusterin, which then induces this CD33 item inhibitory signaling, which then shuts down these protective microglial responses, particularly, as shown here, A-beta uptake and plaque clearance. So the one final question that remains unresolved and is a bit of a fight in the genetics world is whether or not the CD33 big M, which is the common allele, is risky, or whether the CD33 little m is protective. And this is a question that I think isn't fully resolved. But the arguments in favor of CD33M, essentially the wild type allele being risky, is that, as shown here, that protein gets to the cell surface, it binds to AD-relevant ligands, it activates inhibitory cell signaling, and it modulates in a negative way the AD-relevant phenotypes of A-beta uptake and plaque clearance. And in contrast, that little allele of the little m protein doesn't get to the cell surface, it doesn't bind any AD-relevant ligands, and in fact doesn't even have a ligand binding site. And so its impact on cell signaling is at this point really unknown, particularly since it seems to go to the peroxisome, where it's difficult to understand what signaling pathways would happen. And so the question really is the protective effect of this little allele actually simply the absence of the big risk allele? And I think we have some experiments underway which are not complete, but we hope to be able to address that in the next few months. So the final point is, I think, that what this work shows is that the structure enables one to think about small molecules that could potentially block the ligand binding site, although I think that's probably somewhat of a fatuous exercise because the structure of that ligand binding site is not very different from most other Siglecs, and it would be difficult to get a compound that was truly specific. But I think the dimer site is actually likely to be much more specific, and if one could find ways of making small molecules to disrupt that, it would probably serve as a potential inhibitor. And indeed, we know that mutations that disrupt that binding site completely disrupt CD33 signaling. The other important thing is I think it now activates a search for the downstream signaling pathways which might identify secondary signaling targets and also, importantly, I think nodes for crosstalk with other microglial signaling pathways, particularly TREM2, where there seems to be some evidence that these two things modulate each other in a negative way. So let me close by pointing out that this has been a work of a large consortium. The people in blue did the work. The people in black, including the guy at the top with the biggest name, actually did almost nothing. Finally, I'm moving to Columbia, and if there's any interest in acquiring reagents, postdoc fellowships, or PhD studentships, you can contact me at that address or still at my Cambridge address. Thank you.
Thank you very much for your presentation, and time for a couple of questions. Please.
Thank you, really interesting work. So after this amyloid beta plaque and clustering combination is uptaken, are we sure that it is getting cleared out of microglia, or is it accumulating inside monocytes or microglia? Are we sure about that? The second question is that, does the age of PBMC, the patients from which, or the subjects from the PBMC is taken, matters in uptake of this?
So the first question you were asking was what happens to the A beta after it's taken up? And we haven't looked at that ourselves, but presumably what happens is that it is degraded by the lysosome, by other methods. But I don't have data to speak to that, but that would be the assumption. What was the second question again?
Does the age of the subjects from which the PBMC is taken matters in phagocytosis capacity of?
Yep. Again, we have not looked at that, but it's likely that that would be the case. But sex didn't have a difference, and we haven't examined it really enough individuals to be able to give you a firm answer on age-dependent effects. But many of these processes are age-dependent and decay with age.
Guy?
Peter, thanks very much for stepping in, but I'm very glad that I managed to snag you yesterday. I was in crisis because I just had an email from Diego saying that he had woken up very rough and was ill. So, in terms of a question, do you think that clustering, this interaction is specific to clustering as opposed to other sialylated proteins? And do you think there's any kind of physiological role for such an interaction with CD33?
I don't want to leave the impression that the function of clustering is to bind to CD33 as the only exclusive function. That's not the case. We know that clustering binds other things and has other sort of chaperoning properties. So number one. Number two, that it's very clear that CD33 is probably fairly promiscuous in terms of the things that it binds to. Betsy Bradshaw has some work showing CD45 is one of the ligands, but there are likely to be others as well. And I think that's what you'd expect given the shape of the ligand binding site. But it does have some specificity, so it doesn't bind to ApoE, and so it's not a totally non-specific effect. The physiological function, I suspect it's much similar to what we've shown, that it has some role in modulating microglial responses or peripheral blood responses to a variety of sialylated ligands, but in somewhat specific ways. So again, it's not non-specific, but it's not only designed to protect you from Alzheimer's disease. That's probably, or rather, to give you risk for Alzheimer's disease. In fact, that's probably an adverse effect. There's somebody in San Diego who thinks that that there may be some relationship with infectious diseases, particularly things like Neisseria. So,
Thank you for the talk. I was wondering, I know this is the bit that, sorry, up here.
Yeah.
I know this is the bit that puts you into a coma, but in terms of the membranes that are associated with amyloid plaques, is there any knowledge on the sialylation status of those membranes and how they might therefore interact with receptors like CD33?
Yeah, so I can answer you in 2 words. I don't know. 3 words. Yeah, I simply don't know. I don't have any basis to answer that. But it would be an interesting thing. I can't immediately think of a reason why there would be specific cell type membranes. But I mean, it is the cell membrane, so it will have the features of the content of microglia.
Have you checked that these dimers form bigger oligomeric complex that are relevant in the signaling and function?
So you're asking me, is it just a dimer or does it make larger clusters? So
Probably that they need a threshold of signaling enough to promote the function.
Sorry, I didn't quite answer it, but let me just answer as I was going to do. So we know it's a dimer. Whether it makes larger clusters that then give more efficient signaling is an open question at this point. But certainly from looking at sedimentation gradients, etc., it looks like the base thing is actually a dimer, not a large non-specific oligomer. But presumably, if you can get clusters of these dimers at the cell surface, it would actually facilitate signaling in the way that many other receptor signaling complexes work. Does that answer your question? Yeah.
Mm-hmm. And the last question?
You mentioned that I have a very loud voice, actually. Thank you. You mentioned that CD33 clusterin positive microglia, for the want of a better term, were close to A beta plaques. Could I just ask, is that specific, or do you find these cells elsewhere as well, away from plaques?
So you do find these CD33 positive, clusterin positive microglia elsewhere, but they have reduced amounts of that PLA. So it's really a density function related probably to the concentration of clusterin as it seeps away from the plaque. We actually have seen similar density proximity relationship with several other microglial signaling proteins as well. So I think this is a common feature about the activation of microglia around plaques. The closer they are, the more activated the various signaling pathways, and it can be TREM2, CD33, or others.
Thank you.
Thank you.
Regina.